consequence in that you're likely to die, but it's also very, very consistent. So the spread of the predictability of prey having a negative effect on predator fitness is very tight around the line. So the line is steep and the spread of the data around the line are fairly tight. Here's the same slope of the line such that the consequence is pretty great, but the consequence is not very predictable. It's not very consistent. You would expect evolution to proceed more quickly under this scenario than under this scenario. Here's a situation where the consequences are relatively minor. Prey phenotype does have an effect on predator fitness. Once again, in one case, the system is, the response, the effect on predator fitness is relatively consistent. In the other case, the effect is relatively inconsistent. So somewhere in the paper, before you get to the methods, you need to talk about both the strength of selection, i.e. the slope of the line, and you also need to talk about the consistency of that selection, the spread of the points around the line. And so this is just a paper that this one figure kind of explains this in ways that I haven't yet talked about, which might be useful to you. The other thing, if you go and read the paper, and I'm not going to assign this as a required reading, I'm not going to have you write a literature summary or anything about it. When you read the paper, you'll notice some things about how they write about science. You can write about science in an entertaining way that is nice to read and fun to read. For example, they talk about the life dinner principle, which is one of the things we talk about in evolution and ecology, and they use a Warner Brothers cartoon, The Coyote and the Roadrunner, to illustrate this. The fun thing about it is that it's fun. They even cite Warner Brothers, 1952, as their literature citation for the example. It's just entertaining to read in some ways. You can write a paper that satisfies the criteria that are in the specifications for any given assignment, and you can satisfy those things in a dull, boring read that is going to make me want to like slit my wrist because I'm bored to death, and it still passes all of the specifications. But guess what? One of the specifications isn't enriching my life and making me happy. The specifications are about writing well, organizing things well, getting all the information in there that you're supposed to, formatting that information correctly, thinking critically about it, et cetera, et cetera. You can do that in an entertaining writing style, or you can do that in a really dry writing style. One of the specifications isn't make Dr. Kowalski happy. Enjoyment, fulfillment is not one of the specifications. So don't worry about that. They happen to have a way of writing that actually makes it enjoyable to read. Over time, I would hope that you would develop those writing skills, but it's not necessary. What's necessary for you is to satisfy the specifications that I'm giving you. So I think that's the only figure in that paper. So it should be easy to find. Oh no, there's some other figures. It's the first figure, figure 1. Keep in mind that this figure, it has a title, the covariance relationship between predator fitness and prey phenotype. So it's got a figure 1, it's got a title, and then the rest of this is explanation. Your explanations do not have to be this long. You have relatively simple figures, your explanations can be relatively simple. But it's in that description of what's in the figure that you're going to find insight into how to talk about the strength of selection and the consistency of selection in your paper that you're currently writing on. Alexis?
So, judging by that, you're okay with us writing about it in the results of discussion only if it's done well, explained well? It just feels very weird to write about in the introduction, and like I just don't really want to.
The strength of, so you need to either do it at the end of the introduction or you need to do it at the beginning of the methods, or when you talk about the statistical methods, you could do it then, but at some point you need to let me know that you have to be paying attention to two things. You need to be paying attention to the slope, and you need to be paying attention to the R-square value, because both of those things are important for your hypotheses. Because they're both important for the hypotheses, for me, I would logically put them in a little section where I'm talking about the hypotheses. That's not the only place that you could put them. What I don't want them to do is appear in the results having had you not talked about them ever in your methods, for example, as to why are you now talking about them in the results in the discussion. Because when I think of a regression, oftentimes the only thing that I think of as important in a regression, the crucially important thing, is the slope. The R-square value is a descriptive statistic about the spread of the data around the line, so I often look at that, but the statistical test itself is a test of whether or not the slope is different from 0. That's actually what you're statistically testing. And so for some readers, all they really care about is the slope and whether the slope is different from a slope of 0. And
Right,
so
but that thing seems like something that I would discuss when I'm discussing why my results matter, not like in the methods when I'm telling you what I did, also telling you why I did them and why they're important.
if you're gonna, so I would say if you're going to pay attention to a particular parameter about something, you should mention somewhere that you're gonna be paying attention to it so that your reader is aware of it. It's not a failing thing, but it's just nice to your reader to make sure that they're fully informed and prepared. And once again, as I was writing, if I was writing an introduction, when I got to this idea of unconscious versus conscious selection affecting the rate of seed change over the time frame in which agriculture developed, I would want to be framing those hypotheses in terms of both the strength and consistency of selection, not just the strength of selection, because both of those things are important. So I actually would put it formally into my hypotheses, because your hypotheses about what unconscious selection is and what conscious selection is hinge on not just the strength of selection, but also the consistency of selection. So I would put it all the way back there to once again give my readers some idea of what I should be looking for in the methods, what I should be looking for in the results, et cetera, et cetera. Is it required that you do that? It isn't. It would just be kind of better science writing rather than worse science writing. You always want to let your reader in on things early enough that they know what to pay attention to as they go through the rest of your paper. You don't want to spring things on them later, generally speaking. But anyway, at some point you need to be talking about that. I realize that in your version of your paper you talk about it much later. That's fine, you do eventually talk about it, you talk about it in appropriate terms. I would have done it earlier. You do it when you do it, and that's fine. And you also are doing it correctly. Your treatment of it is accurate, so you're fine where you are. That's not how I would do it, but that's, once again, I've now talked to 5 of you, or read, I've talked to 5 of you, I've read 3 of your rough drafts. Every one of you have a differently structured introduction. In all cases, there's nothing wrong with the structure of the introduction as you've written it. Everybody's going to approach it in their own way. I'm smart enough to figure my way through it. Other questions? Questions about what we talked about last time? None? All right, we're going to talk about 4 forces of evolutionary change, and we're going to talk in particular about the theory of evolution via natural selection. Some of this we mentioned last time. We're going to go through it in somewhat more detail this time, because we need to go through some of it in more detail. Yeah, Jackson.
Did you happen to post some notes online for today's lecture, or the slides?
Yeah, it's in, I think it's probably in Wednesdays,
Wednesdays.
because that's when we were supposed to have gotten to this material. But yeah, it's up there. And there are a couple of changes, but they're relatively minor, but I can update it also. As I was reading through, I shouldn't have said it that way. So, 4 forces of evolutionary change, you gave them to me the other day. Mutation. Random genetic drift.
Migration.
Migration. Selection. Awesome. You guys should certainly remember these things long after you have graduated from Williamsville College. I'm going to begin with mutation. So in order to get through mutation, we have to talk a little bit about central dogma, because central dogma is what we use to talk about how we go from DNA to RNA and from RNA to protein. Protein is in many cases the thing that has a role in determining phenotype. So DNA gets transcribed in messenger RNA. Messenger RNA is used as a template to specify amino acid sequence of proteins. Can somebody go to this figure and walk us through it? You all have had cell molecular biology, you all have had genetics, so this should all be within any of your wheelhouses. I'm thinking of a number between 1 and 100. Caleb.
50.
What?
50.
50. Luke.
3.
Haley. We're gonna do this. Jalen.
42.
Huh? What?
42.
42. Logan.
1.
Jackson. Kennedy. What?
That's my number.
99. Okay. Claire.
21.
And Alexis.
76.
Oh, my number was 86 actually, as it turns out. So Alexis gets to go to the board.
I'm gonna do my best. I'm gonna pre-warn you all. I left my glasses in my dorm room, so I can't even see it.
Guess what you have in front of you?
Not that picture.
Or behind you?
All right.
You have an entire classroom full of people who possess the same knowledge
I
that
think
you've
I
been
got
exposed
this.
to, so you can ask them for help if you need it.
The nucleus and the cytosol. DNA being broken apart, and
Do you recall the enzyme that does that?
polymerase.
It's helicase, yep.
Helicase, and then is it polymerase that puts it back together?
It's polymerase that is going to add the RNA nucleotides to that strand of DNA.
Yes, and then here you have rRNA, which is leaving through some kind of membrane protein.
So that's ribosomal RNA and transfer RNA are up there.
Yeah, and transfer RNA is going to like grab these amino acids and become a part of the ribosome, like carrying them into different sites and
Cover that when you get to
together.
translation. Super
Okay.
transcription for now.
So there you have the mRNA with the introns, which are the important part that has to stay, and the exons are not important.
Exons stay.
Exons stay.
Introns
Okay,
get
cool, that's fine.
get spliced out.
And then let's see, so that they go by, because now all the exons are joined together to make the free mRNA. What does that say? What is that, yellow?
Cap.
That's the 5' 7 methyl guanylate cap.
Okay, yeah, and then like a bunch of A's are added to the other side.
The poly A tail, so you get the pre-messenger RNA. That pre-messenger RNA gets what they call post-transcriptional processing or modification in this figure. That's when the introns get removed so that you're left with nothing but the exons.
Yes.
Also, you get this 5-methyl guanosine cap added to one end, the poly A tail added to another end. All that happens in the nucleus before it's then exported out of the nucleus into the cytosol, and Alexis will continue now.
Yes, now it's in the cytosol, all the RNA are in the cytosol, so you have your large and small ribosome subunits, kind of conforming around the mRNA and.
We're at in the messenger RNA.
At the
What's the AUG?
start codon.
That's the start codon, the beginning of translation.
At the start codon, and then it's kind of slowly scooping down, and each time it's bringing a new tRNA with an amino acid that matches the, the
Anticodon.
yes, the anticodon, the triplet, whatever it's inside. It's coming in, joining the amino acids together, and like out comes the protein until it reaches the stop codon, and then there's no more.
Okay. that's a general overview. Anybody have details that they want to add to that? Your vision must be really bad if you
It's
can't
so
see it
bad.
that close.
It's so bad.
We should keep you off the streets and out of cars.
Well, I was driving up here and I almost like head on hit a car because I literally cannot see.
Yeah. Oh my God.
But I left in my room and I was like, let's either go back now. Like as soon as I stepped into the outside world, I was like, wait a second, nothing's clear.
So, when we think about a gene, when you were in high school or junior high, or when did you first learn about a gene?
Probably in high school. But I went to like the worst rural high school.
So, when you were in public school, when you were first introduced to the idea of a gene, you were probably taught that a gene is a sequence of DNA nucleotides that codes for a particular protein. But from here, you can see that genes don't only code for proteins, they also code for messenger RNA, which then goes to specify a protein, but it also codes for ribosomal RNA and transfer RNA, which don't ever produce proteins. They just are used to build structures in the cell that do other kinds of things. And so now we talk about genes as being regions of DNA, sequences of nucleotides that code for functional products. In this case, functional products can be things like messenger RNA, transfer RNA, and ribosomal RNA, but also functional products can be proteins as well. And so broaden your view of what a gene is. Transfer RNA molecules are all unique because they each hook up to a different sequence of codons in the messenger RNA. So their codon sequences are unique, and the amino acids that they carry are unique. And it's that specificity of the sequence in the codon sequence of the messenger RNA and the specificity of the transfer RNA molecules that then allow the messenger RNA to specify a sequence of amino acids. And this is just the basics of central dogma. So another way of thinking about it is we have DNA, the process of transcription accomplished by a complex process that we go into in a great deal of detail in genetics, gives you the messenger RNA. The messenger RNA is then translated into a protein. Note also that there is also post-translational processing of proteins. So the sequence that you get in amino acids out of messenger RNA after the initial protein is formed is not necessarily going to be the same as what you get in the final functional protein. There are also protein folding, what we call chaperone proteins, that govern the folding of proteins to make sure that after they're formed correctly, that they also get folded up into their correct functional shape. So it's a much more complicated process than what I certainly learned in high school and what you learned in high school. But ultimately, we're going from gene to phenotype. And these are the steps that you go through to do that. The thing that sometimes throws wrenches in things is mutation. Somebody give me a good working definition for what a mutation is. So it's change in the DNA sequence. What is the nature of this change? There are different kinds. We'll talk about those in a minute. What's the nature of the change? Is it a directed change or is it a random change? It's a random change, and that's actually pretty important. It is not a directed change. And it can occur in your organism's genome, or it can occur in extrachromosomal DNA or RNA. What do I mean when I say extrachromosomal DNA and RNA? Mitochondrial DNA, and that's animals. Oh yeah, it's animal behavior. We don't have to think about plants.
Good.
What is it in plants? Chloroplasts have their own DNA also. And so it includes not only mutations that occur in the nucleus, but also in these extrachromosomal organelles that are actually believed to be the remnants of eukaryotic cells absorbing bacteria, and then the bacteria co-evolved with the cells and became things like mitochondria and chloroplasts, carrying their own distinct genomes. The endosymbiotic theory of organelle Kennedy's point, these mutations are substitutions, deletions, duplications, transposable elements. I've left one off, haven't I? So you can have a deletion, you can also have an insertion. It's hard to tell which is which, so these are sometimes referred to as indels, which stands for insertion deletion. If you compare two sequences and they're different in one base pair, it's impossible to know whether the one base pair was inserted or deleted, and so we refer to these as indels. Rates vary widely depending on the type of mutation, the gene of interest, the organism, et cetera, et cetera. Substitution mutations, these are sometimes referred to as single nucleotide polymorphisms, single nucleotide polymorphisms, or SNPs. These are of two types, transitions and transversions. What is a transition? What is a transition versus a transversion? Alexis can't help you because she can't see her iPad in front of her.
It's a little clear on the board, but I'm still not getting the point.
You have it right in front of you.
I am gleaning that they're faster than transversions.
Okay, so they occur more often than transitions. Why? Transition is when you substitute a pyrimidine for a pyrimidine, or when you substitute a purine for a purine. Why is that important? What's the difference between the ring structure of a purine and a pyrimidine? What's that here? Oh, who said it?
Logan can say. What was the
Logan.
question?
Logan.
I believe that purines are two ring structures.
Yeah, two ring structures.
Pyrimidines are single 6-carbon ring structures.
Yeah, so pyrimidines have a single 6-carbon ring, purines have a 6-carbon ring bonded to a 5-carbon ring, and so they're bigger in diameter in terms of their total size, and so you typically have who complementary base pairs to whom among these different nucleotides? Adenine, thymine, guanine, cytosine. Adenine always bonds to thymine, that is a purine, complementary base pairing to a pyrimidine. Cytosine always binds with guanine, that is once again a purine binding to a pyrimidine. In this way, we have a 6-ring structure bonded to a 5-ring structure, always bonding to a 6-ring structure. And so, whether it's adenine binding to thymine or cytosine binding to guanine, the distance between the two strands of this double-stranded molecule DNA are constant because you always have an adenine binding complementary base pairing with, you always have a purine complementary base pairing with a pyrimidine.
I think this is what you changed, because the original slides.
Yeah, it's faster. More often than also means faster, so the rate is faster. So when we think about mutation rates are faster for transitions than they are for transversions because when you substitute a purine for a pyrimidine, you get two pyrimidines up against one another. That makes the DNA molecule shorter, not shorter, but not as wide. And one of the things that happens after DNA replication is there are a series of checks on the DNA that the cell does, and one of the checks that it looks for is the DNA molecule the right diameter? Is it the right distance apart? And if it doesn't, it generally goes through and fixes that. And so transitions and transversions have different frequencies. Transitions make up about 2/3 of all SNPs. And transversions make up only about 1/3 of all SNPs. And it's because we have editing capabilities in our cells that basically are focused on making sure that when we do copy our DNA, that there aren't errors in DNA. So, substitutions of the same type of nucleotide versus substitutions of different types of nucleotides. As a result, they'll be occurring at different rates. What is this? Yeah, it's a codon translation table. It basically tells you what are the codons that are possible in messenger RNA, and what do those codons correspond to in terms of the amino acids that that codon designates. What are the important things that you need to note about the codon table? Jackson.
The third letter or nucleotide can vary, but you can still get the same
Yeah.
amino acid.
You want serine, all you need is a uracil and a cytosine, and the last portion, the last position in the codon can be any of the other RNA nucleotides, and you'll still get serine out of the deal. What is this usually referred to as?
Redundancy.
It's redundancy. So you have multiple codon sequences that all code for the same amino acid, and as a result of that, even if there are mutational mistakes that happen, oftentimes you still get the same protein sequence out of a particular gene. As a result of that, though, single nucleotide polymorphisms that occur in the third position in the codon sequence occur at faster rates than in the first and second positions in the codon sequence, because the consequences of a mutation happening in that third position, they don't have any effect on phenotype ultimately. And so the redundancy of the genetic code as it gets translated into amino acid sequences in and of itself will cause different positions in a DNA molecule to be able to have different rates of mutation. So all kinds of things happen that affect mutation rate. These are just a couple of structural things that happen that affect mutation rate. Mutation rates, though, are very, very low. So this is mutation rate from a paper in 2014. This is 10 to the 8th substitutions per base pair per generation. 10 to the minus 8th. You guys know what 10 to the minus 8th is? 1, 2, 3, 4, 5, 6, 7, 8. So that would be 10 to the minus 8th. So that's 10, 100, 1,000, 10,000, 100,000, million, 10 million, 100 million. So 1 in 100 million base pairs per generation. So mutation rates as measured at the whole genome scale are relatively low, depending on where you're measuring these. If you're measuring these within exons, the mutation rate is slightly higher. Whether you're looking at these in disease conditions, whether you're estimating these things using phylogenetic methods, you come up with different rates, but they vary from 1 to 3 per 10 million base pairs per generation. So mutation rates are not terribly high. Now, how many genes does a human being have? How many base pairs does a human being have? Oh, guess what? We know the answer to this question. We'll look it up. What's the size of the human genome? Base pairs. 3 billion. So how many 10 millions are there in 3 million? $3 billion divided by 10 million. 30. What's 3 billion? 1, 2, 3, 2. 3. Thousands, millions, billions versus 10 million.
30, right? Damn, right.
30. So each time one of your cells divides, it has to replicate its DNA. Each time every one of your cells divides, it has to replicate its DNA. So when you were conceived, you became a zygote. Your parents did not know you at this point. You were just a single cell in your mother's fallopian tubes, traveling down. And then all of a sudden, you replicate your DNA and you divide into 2 new cells. In that division, you accumulated 30 mutations. Now you're a 2-cell stage and you're about to divide into the 4-cell stage, so you have to take all of that DNA and replicate it again, but now you're replicating it in both of those cells. And so now you have cell 1 and cell 2 replication on the same genome. You replicate again, you now have 60 accumulated mutations. And on and on and on and on and on and on and on. Okay, what's wrong with this analogy? What is wrong with this picture that I am painting for you?
Considering correcting mutations.
So this is assuming that these are mutations that we can detect, so they're ones that stuck around. What's the y-axis on this figure? It's per generation. So I'm actually talking about growth within an individual, and so actually the rates are not as high. This is across generations, how many get transmitted from one generation to another. But over the course of your development, you do a lot of cell division, and let's say that the rates are lower within an individual over the course of development, you're still going to accumulate quite a few mutations over the course of your development, such that the genome that you inherit from your parents is not necessarily going to be the same genome that occurs in all of your different cells. What's even freakier here is that in recent years we've discovered that when you go through development and differentiation into different tissue types, there are some tissue types where the cells actually just shed portions of their genome that they don't need. And so in some organisms that this has been done, where multiple tissues have been sequenced, you actually find that the tissues within an individual actually differ in the genomes that they have in terms of total genome size, because they actually edit out portions of their genome. This was something I learned about 4 years ago. So this is relatively new information that people were not expecting. I used to teach that once you're born, if you sequence the DNA in any of your cells, all that DNA should be exactly alike. Well, it turns out that's possibly not true. So, but anyway, you possess a number of mutations that come just because when you went from being a zygote to an adult, you have gone through tons of replication. As a result of that tons of replication, you have accumulated a lot of mutations. What's one of the most common forms of cancer in human beings?
Skin cancer.
Skin cancer, why? There are multiple answers to this question.
Sun.
Okay, one is our skin is on the outside. It's constantly being bombarded by a mutagen, which is ultraviolet light. As a result of that, you can expect your skin cells to have higher rates of mutation than cells that are deeper inside. And as a result of that, we get cancers more in our skin. What's another reason that we might expect more cancers in our skin? Not just because it's constantly exposed to mutagens. Jackson.
Skin cells rapidly divide faster than some other.
Our skin cells are constantly, was that what you were about to say, Haley? Yeah, of course. Our skin cells are constantly dividing because we're constantly sloughing off the dead outer layers, producing new layers to replace them underneath. And so once again, our skin has a much higher rate of cell division. It's going through more replication events. The more replication events, the more you can expect mutations to be occurring. So a combination of higher mutation rate because of the presence of a mutagen, plus more cell divisions allowing the base mutation rate to just be multiplied by the number of replications, just makes you more likely to get skin cancer than some other kind of cancer. It's not rocket science, it's just kind of basic cell biology applied to one particular thing. Questions? Yeah, Alexis.
I'm still confused by what per generation means.
So, you are a human being, you are born, you have a particular genome. When you're a zygote, you go through development. If you're a human female, when you're in late fetal stages, your egg cells actually start going through the process of meiosis. At that point, it gets arrested, and it doesn't continue going through meiosis until you start to ovulate them out when you reach puberty. Males develop, they develop gonads. Their gonads are ultimately going to produce sperm. Through that process of development, there are going to be some mutations that accumulate. As you go to then reproduce, you then pass off those gametes to the next generation. You fertilize a new zygote. That zygote now has its own genome. You should expect
That's 30 mutations on?
past 30 mutations onto your offspring.
Okay.
And so if they have if you pass on 30 and, or if you receive 30 from your parents and you already have 30, does that mean it's just gonna compound as you?
Well, so this is how we get genetic variation. Like, this is how genetic variation builds up. So if every offspring produced every human offspring produced has 30 mutations, one would guess that probably most of those mutations are going to be silent, meaning that they either occurred in the third part of a codon sequence such that they don't have any effect on the protein, or they affect the protein sequence in a way that it doesn't affect the function of that protein or whatever. But this is why sometimes you get children who have traits that don't correspond to the traits in either of their parents, in spite of the fact that maybe both of their parents are homozygous for that particular trait. You could actually get a unique trait appearing in an offspring simply because of the accumulation of mutations. You just happen to get the right mutation in the right place, and then had a phenotypic effect that wasn't a phenotype based on a genotype of either of the two parents. So this is just how genetic variation gets built up over time. Like I said, a lot of these, so that's 30 mutations out of 3 billion base pairs. That's not really a lot of mutations, so the odds of any of those mutations being catastrophic in its effect is relatively low, simply because they're scattered all over the genome. And so, and once again, these are single nucleotide polymorphisms. These aren't other kinds of mutations. Yeah.
I mean, maybe I'm just looking at it wrong, but do you know what I mean? Where if, say you have 3 or 4 different generations, the first generation had 30 mutations, passed on those 30 mutations to somebody who already had or to somebody who's gonna have 30 of their own mutations, and then
Yeah, but
they
we
pass
wouldn't,
that 60 on to somebody who's gonna have.
so I get what you're saying.
Are we way more mutants than like humans?
Oh, we're totally mutant. Everybody's totally mutant. So, person A, person B, they get together, they hook up, they produce some offspring, they produce offspring C. Offspring C coming from those 2 individuals basically has 30 mutations. That individual hooks up with person D, they produce offspring E. That person has 30 new mutations, but you wouldn't consider these like added to these.
Okay.
It could be that these things actually are some of these that got mutated also, but it could be that they're all 30 unique mutations, but we wouldn't say, oh, this person has 60 new mutations, because this person has 30 new mutations that are different from
That original
from that individual.
It's not adding on, it's different.
Well, it is cumulative, but we just generally wouldn't count it up that way.
Yeah.
But this is actually how you get, so in terms of the cumulative nature of it, this is how you get change in genomes over time. Because we don't look like we did 200 years ago, or 10,000 years ago, or 100,000 years ago. The human species continues to evolve, it continues to change. If all we were doing was being subjected to selection without replenishing the genome with new mutation, we would get less and less genetically diverse over time. Selection tends to reduce genetic variation. Mutation adds genetic variation back in as it's being selected out. So yes, it is cumulative, but we typically don't think of it that way as we quantify it from generation to generation. But you should be glad that it's cumulative, because that's where we get new variation from. We'll see in a little while how variation disappears under selection, and then we'll be aware of, oh, mutation is a good thing. We oftentimes think of mutation as being an unmitigated bad thing. Why? What?
Sometimes we only hear about that.
Okay, so we only hear about the bad ones. Luke, you were saying something?
It was uncontrollable.
Well, I mean, there are all sorts of things that are uncontrollable that we don't necessarily think are bad. I'm not in control of the roller coaster, but I might perfectly enjoy riding on it.
I mean, like, somebody is in control of the industry.
An algorithm in a machine.
Sometimes it breaks down.
Sometimes it does. Sometimes it does break down.
Sometimes it flies off the tracks.
So we hear about the bad ones. Why do we hear about the bad ones?
Because we don't know about the good ones.
Well, we know all about the good ones.
If I have some good mutations, I've never met them.
You have 5 fingers, 5 toes. You have a backbone. You have skin.
It's because they're normal.
Yeah, but that doesn't seem like a
It's 'cause they're
mutation
normal.
that like my mother passed to me.
No, but it was a mutation that some organism passed to some other organism way back in evolutionary history.
Like, well, like mutations that cause like deformities or disease or.
We think about the mutations that cause deformity. So, Alexis, you have a mutation that gives you 5 fingers, and that mutation of a gene is different than the mutation that gives some other people 6 fingers. Well, exactly. Those are just 2 different variants of the same gene. Which one is a mutation depends on which one you determine is wild type. And determining wild type is kind of arbitrary. It's just whatever's the most common thing that is out there. So one way of thinking of mutations is it's just another variant of the gene. Those variants can have no effect. They can have positive effects. They can have negative effects. You are the accumulation of successful mutations. You are the accumulation of mutations that didn't kill you or your ancestors. So we think about mutation as being a bad thing because sometimes the mutations that we observe are mutations that take you from being successful to being less successful, the things that go wrong. But because so many things have gone right over evolutionary time, you are here as a product of successful evolution. Your parents were all capable of reproducing. My parents were capable of reproducing. So at some point, they were fully functional in an evolutionary sense. All of the genes and all of the different mutations of those genes that made them capable of reproducing produced me and you and everybody in the room. So we are the accumulation of a lot of successful variants of genes. So when mutations come up, chances are they're either going to be silent or they're going to be negative, or they're going to be positive. Those are all the choices. They're going to be silent, they're going to be negative, or they're going to be positive. Are you going to notice the positive ones? Probably not, because they're probably going to be subtle positives. We're not going to go from not being able to fly to fly as a result of a single mutation. But what we will notice, we'll notice the negative ones. So the negative ones are more noticeable because they're coming into a background of a lot of evolutionary history that has made us be the accumulation of lots of successful variants. We won't notice the positive ones because most mutations are not mutations of large effect, they're mutations of tiny effect. And so we're going to notice typically the negative ones that are negative ones of large effect. It doesn't mean that other kinds of mutations aren't happening, it's just the ones that we notice and pay attention to are removal or deletion of either single nucleotides or larger segments of genetic material are referred to as addition, deletion, mutation or insertion, deletion. This is where the term indel comes from. These things result in a frame shift. What do I mean by frame shift mutation?
Just three base pairs.
So you have this codon structure of messenger RNA molecule. The amino acids are designated by 3 sequences of the messenger RNA. When you delete a nucleotide or add a nucleotide in there, it shifts the codon structure. And so if this is the wild type, this gets converted into this messenger RNA molecule. This ends up being the amino acid sequence that is coming from this messenger RNA. If you add in an extra adenine here, it disrupts once again, the codon sequence, and this lysine gets converted into a stop codon, so you now have a much shorter amino acid sequence than you had before. If you, that's an insertion. If you have a deletion, you get that out of there, you're missing that uracil. It gives you the same first 2 amino acids, but then every amino acid from that point on will be altered. And the problem with insertions and deletions is that they have these catastrophic effects on the ultimate protein sequence that gets specified, because they've disrupted this 3 nucleotide base sequencing that is the codon structure of messenger RNA. And so an insertion or a deletion, if it's at a single nucleotide, it can have much greater effects than a transition or a transversion, because it affects not only the base pair where that insertion or deletion occurred, but it affects every base pair from there on down the DNA. It affects that gene. If it occurs in a place that there's not a gene, it affects that intron and then all of the exons and introns all the way down the remainder of that chromosome until you get to the end. And then you get to start over at the beginning of the next chromosome, because we have our DNA from the next chromosomes. Insertions and deletions have a much greater effect than single nucleotide polymorphisms. Okay, we talked about random genetic drift last time. I want to provide more today. What did we say about random genetic drift on Wednesday? Caused by random deaths. Where does random genetic drift seem to exhibit its effects most visibly? Small populations. We have a small population here, right? How many individuals?
12.
Or 12 individuals, which means we have 24 24 alleles. How many of these alleles are the dominant allele? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 12 of the 24 are big A allele, 12 of the 24 are little A allele. What is the frequency of the big A allele? What's wrong, Caleb?
I was counting. No, I was counting each one was in that.
Alleles.
Yeah, I did it.
Frequency of A, which is P, is 0.5. Frequency of the little a, which is Q, is 0.5. Kennedy, flip the coin for me. If we get a heads, you live. If we get a tails, you die. Yay! Yep.
We just died. You died.
Oh, sadness.
Yes.
Sad. Oh,
Died.
oh my God, you're terrible.
Died.
Oh, yes. It's a bloodbath.
Died. Died.
With the eye.
I don't think that makes a difference. Died. Died.
One survivor.
The odds of this are getting more and more unbelievable as time goes on.
Die.
No.
I'm gonna illustrate.
Yay, live!
Live.
Yay, live! Okay.
Like,
to random death.
What is the, what's the first one on the middle?
And well, she was flipping a coin. She was doing the very like quintessential example of random. So we went from 12 individuals to 3 individuals, which means we have 6 alleles. 1, 2, 3 of those alleles are the big A allele. 3 of those are the little A alleles, so the frequency hasn't changed. Okay, that happens sometimes. Give the coin to someone else.
Sorry.
You're destroying my point. All right, heads or tails?
Heads.
You live.
Heads.
You live.
Tails.
Sad.
Heads. Tails. 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 Heads. Tails. Heads. Tails. Tails.
Okay, so now we have 1, 2, 3, 4, 5, 6. No, not 6. That guy's dead. 1, 2, 3, 4, 5. So we have 5 individuals, so we should have 20 different alleles. 1, 2, 3, 4, 5. 5 out of the 20, which is 1/4, are the big A allele, which means 15 out of the 20. Is that right?
Wouldn't you be at a 10?
Oh, I'm sorry. It's out of 10, rather.
So you're still the same.
I'm still at 50-50. What is wrong with you people?
Hey, it's 50-50. It's a coin. I don't
Who's
know.
next?
I'll do it. Get it to Alexis.
I'm literally just like chilling.
Give that coin to someone else.
What's it, why does that say 12, 12, and 24, 12? I'm
There
gonna
are
say
12 individuals, 24 alleles. 12 of the alleles are this, 12 of the alleles are that.
I'll do it.
Caleb, do me right here.
Tails. Tails.
Come on, kid.
Heads. Tails. 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 Heads. Tails. Tails. Yeah, heads. Heads. Heads.
4 individuals left standing. 1, 2, 3, 4, 5, 6. 6 and 2. There are 8 alleles total. 6 out of 8. 2 out of 8. Finally, so in these 3 runs, we flipped the coin. In 2 of the runs, we didn't change the allele frequency from one generation to the next, but in the other case, we changed it from a 50-50 mixture to a 1/4 mixture. This is the nature of random genetic drift. Allele frequencies within populations change. It does have a head and a tail line. Allele frequencies change not because it had anything to do with the genotype that these individuals had or their phenotypes. It had everything to do with just random chance of perishing. Going back to wearing a garb, walking along the sidewalk and having a piano fall on your head. This is the nature of random genetic drift. I don't have this on the list of things that you need to have at your disposal, but back in evolutionary ecology, we introduced you to this program called Populus. And if you still have it on your computer, you can go and find it. If you don't, it's not that big appeal. Um, Oh, I don't have it anymore because my computer died. Anyway, there's a program Populous, and one of the things that you can do in there is you can simulate. Oh, can I borrow your computer real quick?
Yeah. It doesn't have USB though.
Yeah, that's fine.
It sucks. You have to use it for
I'll just show people
Yeah.
up here.
Oh, man.
Sucks. They said that when I bought it, they were like, oh yeah, all the USBs will be that whenever you know
Yeah.
third year in college, and none of that.
So on this
Sales guy.
thing, you can vary population size. I'm gonna put this down to 5 individuals, and then what it does is it tracks multiple populations. And let's see. So, and what you see is each color is a different gene, and you can see that these things are blinking out over time. They either go to fixation or they go to extinction. This is the probability of this is the frequency of the dominant allele for 10 different loci. And so by 15 generations, all of the loci have gone to either fixation, meaning 100% of the dominant allele, or extinction, 0% of the dominant allele. By the time you get out to 15, if you increase the population size to 20, for example, you're now out here at 60 generations, and a good number of these things haven't gone to fixation, either fixation at the dominant or fixation at the recessive. And so as you increase sample size, as you increase the population size, let's go up to 100 now. You see that, once again, more and more of your populations are not at fixation. Sorry, Alexis, you cannot see this. Bring your glasses next time.
I'm sorry.
And so you can get on here and play around and say, okay, what is the effect of population size on fixation, fixation is either P going to 1, meaning that it's all the dominant allele, or P going to 0, which is all the recessive allele in the population. And you'll see that the larger the population is you're dealing with, it takes longer and longer for you to get to fixation. And this is the nature of drift. Migration is really straightforward. Individuals leave one population and go to the other population, or leave the second population and go to the first population. When we transfer an individual from one population, if they happen to be a homozygote, they've removed 2 of the little a alleles from that population. It's changed the allele structure of the first population, and it has added those alleles to the allele structure of the receiving population, and as a result, those 2 populations have different allele frequencies than they had before. And that's the very definition of evolution. So migration moves alleles around. Selection we've introduced to you in the past. If we have a quantitative kind of trait like height, we begin with a population that is normally distributed. If we select against one extreme of that population, in this case we're selecting against short individuals, the population after selection has shifted over, the average phenotypic value has shifted, and delta Z is the evolutionary response to this selection. We can also have selection that acts on both tails of the distribution. Selection equally weighted against short and tall individuals. The population after selection has contracted, but the population mean hasn't changed because the selection was symmetrical. This is stabilizing selection. And then we can also have disruptive selection, where selection acts against the average phenotype, not acting against the extreme phenotypes, and you go from a unimodal distribution to a bimodal distribution. Back in evolution and ecology, this is what the Darwin's finches are going through, where medium-sized finches don't do as well as large and short-beaked finches under periods of low food availability. So we have directional selection, we have stabilizing selection, and we have disruptive selection. These are the three forms of selection. Do these look familiar to you? Yes. What are they? They're, yes, they're graphs of what?
Oh,
The bean selection, are these your graphs? No, these are the 2018 figures. How do you know that they're not yours? Okay, so the F statistics are different. Same pattern. The slopes are slightly different, well, a good deal different. Yours is 0.0015, and yours are, one of the reasons is that we did lot mass rather than average bean size in 2018, so that's one of the reasons that the slopes are different, because of the magnitude of the numbers are different. Once again, weak selection, strong selection. Is this directional selection, stabilizing selection, or disruptive selection? Why? You're selecting against the small ones. In one case, you're selecting against the small ones in an unconscious way, and in the second case, you're selecting against the small ones in a conscious way. But it's not, it's one-sided selection. You're selecting it, and it's one of the extremes of the phenotypic distribution of seed size, the very definition of directional selection. It's 135. Fisher's Breeders equation. We talked about this in evolution and ecology. We will revisit it again on Monday. If you have not read Arnold's paper yet, read that for Monday. We will pick up here. Do you have any questions?
Can we put off chapter 2 for a minute?
Yeah, because we'll be finishing up. We'll be finishing up natural selection, which I think is technically covered in chapter 1. We'll get to chapter 2 probably towards the end of class on Monday. So if you have time, go ahead and start reading it, but we won't get very far into chapter 2.